Hi everyone, this is Amanda Majorano, and I'm here to talk about a study we have done in Nelore cattle. The objective of this study was to detect large polygenic genomic regions in two subpopulations of Nelore animals raised in Brazil. The first subpopulation was selected for racial characterization, mainly morphological traits, and we call it this subpopulation as PO. And the other one was selected for economically important traits, mainly growth traits, feed efficiency, and cassette trait. We call it this subpopulation as SAIP. As we use it sequencing data, we have this amount of information from the allelic SNPs, and they were located on the bovine autosomes. The Ray and Kirkhian FST estimator was used to compute the allelic differentiation between the two subpopulations. We applied a minor allelic frequency above 0.01. The segment FST function was used to identify polygenic selection. Basically, what this software does, what this function does, is to define a start and an end for regions, and it considers only SNPs that are above a threshold. So this threshold was defined based on the percentile of FST values, and this means we used a threshold above 0.3. At the end of our analysis, we got that the FST significant values were spread across the genome, and regions were found in different chromosomes. In total, this amount of highly differentiated genomic regions were found, and we used the newest cattle reference genome annotation file to search for genes, and these regions, these polygenic regions overlapped with 271 genes in total. We highlighted here 3 different regions. The first one located on chromosome 5. This region encompassed many genes and novel genes. For example, this gene here was related to growth and novel score in another study with Nelore cattle. And it was important to comment from this gene here on chromosome 21. It was related to feed efficiency and carcass traits in Nelore cattle in other study. And also the region on chromosome 23, it was important that this region was identified in our study because this is a highly polymorphic region in cattle. This is part of the bovine MHC. And this region that we found on chromosome 23 encompasses many genes. We highlighted here a variant located with the region of BOLA DBR3 gene because this variant presented the highest FST value within this region, and is close to a QTL reported in cattle. This SNP is higher in allelic frequency for the PO subpopulations, so it means it is being selected in this subpopulation. The results we found here will contribute to our knowledge regarding genes and variants that have an important role in complex traits selected not only in the PO subpopulation, but also in the SAIP population. Thank you for your attention, and if you have any question, feel free to send me an email. Thank you.